There are over 20,000 genes and 37 trillion cells in the human body. To keep everything under control, eukaryotic cells must use a variety of different methods for genetic regulation. But what happens when one of these regulatory mechanisms go wrong? Well, like an orchestra without a conductor, too much or too little. For example, a sign of polycystic kidney disease is the presence of high levels of protein F. Most cases of polycystic kidney disease are known to be caused by mutations in the PKD or PKHD family of genes. When functioning normally, these genes facilitate communication between the cytoplasm and nucleus of a cell to promote normal kidney development, organization, and function through protein activity. So, what are the possible mechanisms for the overexpression of these genes? There are two possible hypotheses. First, insulator regions located up or downstream from the target gene has experienced a missense mutation resulting in the respective insulator binding protein from being unable to attach. Insulators are a class of non-coding cis-regulatory elements, meaning that they are located close to the target gene. Their primary function is to recruit insulator proteins to separate domains on a chromosome. Transcription requires the assembly of the basal transcription apparatus and the recruitment of transcription activators at the regulatory promoter and enhancer region before RNA polymerase II activity can be fully activated at the core promoter. Enhancer regions can be located some distance from the core promoter site. These enhancers are capable of interacting with a wide variety of promoters. This is why insulators are so important. In short, they prevent the wrong enhancer from interacting with promoters that are separated in a distant domain. Without it, it would be as if several conductors are attempting to direct the orchestra at the same time. Therefore, gene expression could potentially be upregulated. Second, the inherited disease allele produces SR proteins that bind to alternative splicing sites of the pre-mRNA that are not normally used, resulting in a mature mRNA that has an enhanced poly A tail. When transcription is terminated, the pre-mRNA strand is cleaved, and processing immediately begins, including the addition of a poly A tail at the 3' end. An additional characteristic of eukaryotic pre-mRNA is that it can have multiple splicing sites, allowing one strand of pre-mRNA to code for multiple proteins depending on how it is cut. In normal functioning cells, a normal production of SR proteins regulate what splice sites are used or silenced. However, in malfunctioning bodies, an overproduction of the wrong type of SR protein can have consequences. When this happens to the poly A tail of pre-mRNA, it is referred to as alternative polyadenylation. This poly A tail helps maintain the integrity of the resulting mRNA molecule after it has been fully processed. A mature mRNA molecule can then be translated into proteins by ribonucleases. However, every time it goes through translation, the poly A tail becomes shorter. After multiple translations, the poly A tail is no longer able to sustain the stability of the mRNA, allowing it to degrade. However, if the poly A tail has been altered to enhance stability of the mRNA, then ribonuclease will continue to create protein long after its intended use. Although everything seems well at the allele, the improper splicing of the pre-mRNA upregulates protein F production.